now reintroduce Randal Koene, who gave an excellent tutorial yesterday. And today, Randal is going to give the second keynote talk, which is about whole brain emulation. So Randal is a neuroscientist rather than an AI guy per se, and one of the reasons we invited him is not only because he's an outstanding researcher doing interesting stuff, but because I think it's important to mix up disciplines in our attempt to make thinking machines. And I'd be happy in succeeding years to have more contribution from engineering, philosophy, psychology, all sorts of allied disciplines, as well as computer science. And his talk yesterday on reinforcement learning in the brain as compared to reinforcement learning algorithms, I thought was quite inspirational. And looking at the details of how we may eventually be able to make AGI systems closely modeling what the brain does, while it's not the research direction that all of us are taking, I think there's a lot we can learn from that. So Randal is now the director of a neuroengineering department at Technelia, which is a major company in Spain, and he was at Boston University when I first got to know him in the US. I'll turn it over to Randal to tell us how we will one day upload ourselves into computers.
Okay, thank you very much. So this talk is actually, I would say, a little bit lighter even than the tutorial that I gave yesterday. I'm posing more problems, asking more questions, and not giving as many examples of any kind of possible answer. Still, I hope it's interesting, especially regarding what Ben just said about this cross-fertilization of ideas across different disciplines and trying to look at what we can learn from neuroscience that you might be able to use in AGI. Interestingly, yesterday, in the discussion with Richard Sutton after his talk, one of the things that came up right away after digging into a few issues with what may be possible, may not be possible in AGI, is the question of how many details, what kind of details do you need to know about if you want to learn from the brain how intelligence works in the brain, if you try to implement this in an artificial structure. So the question becomes, do we look for one function that can explain how you can do intelligent things, or do we try to look at a lot of different implementations that are very specific to the task at hand? And of course the brain is an evolved structure, and that means it comes with a lot of baggage. In the case of the human brain, we have pieces you can definitely still recognize from our reptilian ancestors and before. So there are some parts that work quite differently than other parts in the brain. There are structures that are very different. Sometimes there are very regular structures with multiple layers, and sometimes it's very chaotic, and sometimes it's more a sparse network with a tree structure, and you have all sorts of different anatomy in the brain. And also the neurophysiology, so the way that the bits and pieces inside these structures work, differs from location to location and from task to task as they're all employed in different tasks. This is a natural consequence of the way the brain evolved. It's not necessarily telling us that we need to do the same sort of thing if you're trying to implement an artificial brain. You might do that quite differently. But it's important to keep in mind if you're looking at our brain or the brains of animals as inspiration. So what I want to talk about today, whole brain emulation, issues of scope and resolution, and the need for new methods of in vivo recording. The reason I'm giving this talk is because in previous previous occasions, I've often focused on looking at the structure of the brain and learning from that, and the importance of in vivo measurements seem to fall a bit by the wayside, and I hope to bring that up a bit more today. So first I'd like to just throw out a few definitions, not the best ones necessarily, but useful ones about what is whole brain emulation and what's the rationale behind this field of research, and then some of the leading researchers in the field, or the leading points of research, directions that are being taken. Then, secondly, I'd like to look at the problem of scope and resolution, to look at what sort of data you need for brain emulation, how we can discover the resolution we want to find, that we need to use in order to replicate activity in the brain, or replicate mechanisms of the brain, and how you can do in vivo measurements, or what sort of in vivo measurements would be necessary to find the resolution. And then, because that is, of course, the question, is how can you find that real data? We need techniques to do this. And there are reasons why the techniques that we have now are not sufficient. And there are some good prospects in certain directions of research that I would like to emphasize about next generation in vivo recording techniques. And then I'll just wrap up with a couple of comments, general comments. So, definitions. Well, what is whole brain emulation? One way to define it is to say it's taking the functioning mind, what we consider the mind, and that again requires a definition, and implementing it in some other substrate other than its biological substrate. Now, when you're thinking in general terms of a thinking entity, then you could say, oh, well, this definition also applies to AGI. It's some other substrate, and you've got something in there that you would equate with perhaps a mind. But here I really want to be constrained to the human mind, so our minds, what we consider us. And how would you have something like that function? So have all the same functions, the same memories, the same operations, but not in its biological form. And there are many reasons to do that. One of them, and an obvious one for all of us researchers, is the fact that the biological substrate is very hard to access. If you want to do anything with it, such as fix it, someone had an accident, it's very difficult to do. If you want to do something with it to augment it, because perhaps you would like to be able to reference Google automatically from your brain or something like that, it's also very hard to do. Or if you just want to understand what's going on moment to moment in some part of your brain, as long as it's in this biological substrate, it's very hard to handle. So for researchers, to have something that works like the brain, but is in a different substrate, is of course really interesting. And that, it's not a novelty to say that, because that's the origin, of course, of computational neuroscience, and even of any type of modeling. Now the other definition is a bit more precise: a mechanistic re-implementation that enables predictions of active state and behavior at t plus delta t with acceptable error. So here the idea is really, how can we take what's in our brains and have something that emulates how that functions so well that you could actually predict 1 second in the future what's going on in that, what would be going on if this were the biological brain, what would be going on in that mind at that time, with some acceptable error. And that's another topic. By the way, I just want to say that this is, although it's not a tutorial, lectures generally also allow people to raise their hands and throw up a question or something, so I don't mind if people interrupt me. That's fine. Reasons why you would emulate. I've mentioned a few already, but let's go over some again. The system is known to work. When you're dealing with systems, well, you're trying to create systems in AGI, well, you're making a lot of hypotheses. Yesterday we saw numerous assumptions and ideas about how would this work, and maybe it doesn't work this way, and then criticizing one technique over another. That's fine, but we do know that there is one system that does work, and that's the brain that we have. And not just ours, but animal brains and insect brains. All of these, to certain extents, work and do things. So it's very useful to look at them, because when you have a working system, well, you can find out why it works, and then you can try and replicate that, or come up with a better way to do it. So, this gives us a chance to properly ground our assumptions and hypotheses. That's something that's often missing. Not just in AI, where you often start top-down or bottom-up and try to fill in the middle along the way and making a lot of assumptions, but also in neuroscience, many of our models and our theories and hypotheses are really very loosely grounded. There is not a lot of hard science that's fixing it all in place, and that's missing. That's something that in chemistry and physics we see that a lot more, and we would like to see that in neuroscience as well. And then there's the ability to restore function. So this doesn't only apply to science and trying to understand the brain, but what we're trying to do here in this field of emulation is also applicable in, let's say, engineering and medicine. So, for instance, neuroprostheses. If you want to create a neuroprosthetic augmentation or a neuroprosthetic replacement part, say for someone who doesn't hear anymore, the cochlear implant is always a popular example. Well, that's a case of trying to emulate what's going on in the original part of the brain that's not working so well, or the sensor, or some piece right in between in the perceptual area. And then, then on my last point here, human-derived AGI, if you're going to be engineering things, and if you're going to be making emulations, you could call such an emulation a human-derived AGI. When you have something running in a different substrate that does what a human brain or an animal brain would be doing, you have a type of AGI. Now, why consider the whole brain? I get that question a lot, because it's a big thing, and it's easier to study parts than to study the whole, or to even emulate the whole. And I agree. I think that many times we have to go down to pieces and look at those in order to understand what's going on and to make our first models, let's say. Not just the first, also the second and third. But eventually there's a good reason to create a whole brain emulation. One of them is that the brain is modular, and yeah, it goes back to my argument about evolution, but there are also other reasons why you would make a modular system. And all these modules depend on one another. So you can't only say, okay, we have a system with so many neurons that are in one network, and you give it an input and output, but there are actually many networks, and those networks work together in different ways, some at different rates, different time scales involved. Some are responsible for storing our memories long-term, and some are responsible for picking up events rapidly and just storing them for the moment to be filtered later and find out which things to keep and which things to throw away. So there are a lot of modular systems in there. And if you really want to understand how we think, how we work, then you would have to look at the whole thing, all the modules together. I agree it's completely ambitious, overly ambitious perhaps, at this time, but I think it's worth looking at, and honestly, I have nothing better to do for the next 40 years, so. Now, which parts would you consider significant? Because when we say whole brain emulation, some people think just look at your cortex. Others think you have to take the spine and you have to take everything that's in your head. And some would say, well, no, you need a body emulation as well. And some would say, oh no, you have to put an entire virtual environment around that. So the question is, what is a whole brain emulation? Which parts are the whole brain? And that's something that I won't look at today, but that is also important to consider. For instance, I think I wouldn't necessarily require legs to be part of it. A simulation that is really a close simulation or emulation of how the legs work, in anatomy, etc. I wouldn't require that. Even though I would like to be able to experience mobility and understand how that works in an emulation. Vision, and this gets trickier. If you throw out vision, you're really tampering with something that I think is very important to the way that the brain manifests itself, that the mind, manifests itself and the way that we think. Although, of course, blind people may disagree. No new episodic memory. Let's say that we throw out the hippocampus. I would say that's not really important because we're more interested in a system that already has learned knowledge, long-term memory, and we want to see how it behaves. Okay, well then you have a system that does that and it doesn't accept any new events. Maybe that's also useful to look at, but to me that's not a whole brain emulation. Initial partial studies and whole brain may not constitute conceptually significant different problems. What does that mean? What I'm trying to say is that you can scale up. And if we at first do a whole brain emulation of Drosophila, just to get an idea of how to do this, and you have that at the scale of these little flies, I think that's quite worthwhile because many of the same principles will apply across different species and will apply even for humans. Also, if you take just a portion of the human brain, let's say you're emulating how the hippocampus, to go from the hippocampus again, because that's one of my areas of specialty, works, then yes, we can learn a lot of basic principles about how neurons operate together, what things matter at which level. You know, what about dendritic computation? Is dendritic computation important? Things like that. And then you can scale up by applying this to other areas. And scaling up is sometimes, sure, it comes with its own problems, but sometimes easier than, you know, than the original work of trying to figure out how all the basics work. So whole rate emulation may sound like an enormous task, but going from 1 billion to 100 billion neurons may not be as much of a challenge. So it's complex, but the neurophysiological challenges may be similar. Now, why? Okay, this is another issue. We've been talking about whole brain emulation as if this is something that you could just do for any brain, and then you've got some sample system sitting there. But what does it really mean? Subject specificity. Are you going to emulate the operation of a particular brain, one that has a certain anatomical structure, certain learned memories, etc., or are you going to emulate a general system? And we'll see some examples of both in the next the work stuff I'm going to show you on the leading research. I think there are reasons to consider specimen-specific or person-specific emulation. One reason is that we're not really sure at the moment which aspects of anatomy, physiology, and the dynamics of the brain are generalizable across all of us, so that I can just take this general model and then tweak it a little bit to make an emulation of X or Y, and which ones need to be adjusted to these years of, I guess, modification that are going on from the moment we're born, and even from the constitution of our particular infant brains based on our DNA. So these differences may be more severe than some hope. And so there's certainly cause to at least investigate subject-specific brains to find out what are these differences before jumping to any conclusions. Another good reason to be subject-specific is, this next one really ties into the previous one, that identifying what are the significant aspects of circuit function may require that we at least analyze it in a subject-specific way. But then there's also, again, this application outside of the science itself, but to the engineering or medical approach, which is if you want to make a neuroprosthesis. Neuroprostheses, like any prosthesis, are naturally meant to be adapted to an individual. So, and I'll just jump ahead a little bit because this will come up, but Ted Berger, for instance, is trying to make a neuroprosthetic for the hippocampus. There we go again with the hippocampus. And one of the problems, actually, with what he's doing right now is that the way that that model is built is not subject-specific. Or rather, it may be built according to one subject, but then used in another. And there's no reason that we can assume right off the bat that this will work. I mean, why would one circuit like that, if you implant it in another system, why would it work? After all, the whole system, this whole multi-modular system, has developed over years and years together, so that you may not get the right input-output mapping, and perhaps even what's inside the little black box that we're calling the hippocampus there may be doing something different in person A or person B. So if you're doing it as a prosthetic, then you really have to think hard. How much can we generalize, and how much do we have to do in a subject-specific way? Now, when you're talking about sensory systems like cochlear implants, then perhaps you can get away with doing it in a more general fashion, because the core system can adapt to those kinds of changes. Our hearing changes over time anyway. I mean, we change how well we hear when we're children and how well we hear when we're elder. So that's, we can kind of cope with that. Okay, some leading research. One that you've probably heard about many times, Henry Markram and the Blue Brain Project in Lausanne. Well, there are two main parts of this project. One was to develop a detailed model of the neocortical column. And this is similar in some sense to the kind of mini columns I was talking about yesterday. But in this case, we're talking about a column of 60,000 neurons in the human brain. So 2 millimeters tall, 0.5 across. This is the representation of the complete neocortical mini-column simulation, which was completed, according to Henry, in 2006. But it's not 60,000, this one is 10,000 neurons because it's based on the rat brain. So here we have another case of going and saying, okay, we can scale up. You can learn it from one system, the rat, and we can then scale up to the human if we want to do the human, because the eventual goal, indeed, is of course the human brain. And this shows you very little or absolutely nothing. Henry has beautiful animations that display what's going on inside these simulations of the neocortical column. And I can't show you these here because I didn't have time enough to ask them if that was okay, but they're really very nice. It's based on architectural principles. So where are the connections generally? What are the long-range connections, short-range connections? What connects with what? Which type of neurons do you see? What is the morphology of the neurons? So it goes down to that level, certainly, and to the molecular level in some sense, because he knows which neurons are using what types of transmitters, and so forth. He's done years of research before characterizing all of this kind of information, and then brought in a whole bunch of students, as well as some very nice sequencing robots and patch clamping robots, to quickly go through batches of data and discover more about what is in this neocortical column, what kind of data exists there. Now this gets us almost to the bottom line here, even though I haven't talked about this yet. What I've said so far is, okay, he's collected a whole bunch of data. First of all, it was in the rat, and secondly, it was done with a whole bunch of different rats. So the data that he has is building a construct. This is not anyone's, not even any particular rat's neocortical column. This is a general neocortical column. Yes?
So I guess a lot was learned in building this model, but once the model was built, what sort of things have they learned from the running simulation?
So far, I think that most of what they've learned is more based on looking at individual details inside the neocortical column and doing experiments on, for example, the strengthening of synapses between particular regions and whether or not they can operate as subsystems, as microcircuits that carry out tasks. I'm not an expert on what Henry is doing with this neocortical column, but I know that he has right now the simulation that's based on the neuron simulating environment, running on a Blue G computer, and that simulation is basically always on. And the students and postdocs, they come, they have certain challenges in mind, and then they can pose those, put those into the simulation and run part of that. So they've got one simulation, one big simulation, and they're doing work with that. Some of the work is kind of still trying to fix the final bits and pieces of putting together the neocortical column. For example, there was some difficulty in determining which synaptic connections were wrong and needed to be pruned. And so there was some interest in looking at, for instance, developmental ways to find out where these connections would go. And that tied in with some of my work at the time.
And how
Yes.
did they choose their inputs?
So how did they choose the inputs?
Inputs on this? Because as I understand, it's difficult to catch from the in vivo real
Yeah.
inputs.
Well, you could do two different things, really. I mean, they've done a lot of experiments in vivo where they did some were on local connections, very close connectivity, but also long-range connectivity where they would stimulate axons in one area or neurons in one area and then see what happens in a different area. So that's how they figured out a lot of that information. But then in the model, they really used, from what I know, two different approaches. One approach is that you sort of replicate these kinds of studies that are really very small scale, where you do a little bit of stimulation here and then some detection over there. And the other is spontaneous activity, where you have random variations in membrane potential and causing some neurons to activate, and then you get spreading activity. So you can observe things like oscillations that happen in the system. You can observe the flow of information from one cluster of neurons to another, things like that. Yes?
Yeah, but there's also been some controversy regarding these experiments, I believe, because first, it is a construct, it's not any particular mini column, right? Then it's kind of a free, it's free to test it the way you like, right? So how you test it will determine the correctness of your system construct, but it's hard to do that because it is a construct. And second, Marvin Minsky commented that because they didn't choose the right inputs, that there was no way to test if it was correct. But my understanding was that their tests were qualitative, not quantitative, and that's where it seemed lacking.
Well, I think you're right, and I think that doing real quantitative tests on it is difficult, in particular for the reasons what I mentioned before. Why do open emulation? Why do it subject-specific? Everything you've just said. The problem is that you've got a system with 10,000 neurons, very complicated neurons, because the morphology is detailed. This means you have a highly over-parameterized system. You can put anything in that you want and make it do anything if you just tweak the synapses the right way. So what it does, and then saying, here we have a successful experiment, it's dangerous to make those claims because you are in control of what this does, and the system is basically a general purpose computer.
Yeah, there isn't a control group in the experiments.
Yeah, I want to comment on this briefly because I've talked to Henry Marker about this a bunch, like in the last 6 months, trying to understand for myself what he actually did and what it means and what he actually plans to do to make the whole brain simulation out of it. So I think whoever said they learned the most in building it was probably right in the sense that in the course of trying to make the simulation work, they had to go back and kind of rewrite a bunch of the basic differential equations for the membrane potentials inside the neurons, which was quite valuable. And Marker and his colleagues had some papers in biological cybernetics on that. So it's just in the course of building it, they realized some of the underlying equations weren't right. And in terms of qualitative testing, I think a better way to say it is they showed that his simulated column had many statistical properties in common with a real column. So I mean, they did mathematical quantitative testing, but they didn't show that their simulated column had a detailed functional input-output behavior the same as a real column. They showed they had similar statistical properties of output based on similar statistical properties of input, which means something. So what I've talked to Markham about personally is actually doing some work aimed at taking one of his columns and making it emulate a column in detail with the precise functional input-output behavior. But of course, even if you could do that, it has a complaint, well, maybe you're just overfitting because there's so many free parameters in the column. It doesn't mean that the real brain works that way. And I also think there's a spin-off of the Blue Brain project now, which still has the Blue Brain name attached to it, which is aimed at going more fine-grained and doing molecular level simulation. And I think he does think you need to go to that level in order to do a detailed whole brain simulation, even though he didn't go to that level in the previous Blue Brain projects that are more famous.
Yeah, I think that kind of thing is really where this work is very valuable, because even if you're dealing with a general system, you can explore all those basic questions, all those fundamental questions. Then trying to say something about, okay, well, this is the neocortical column from rat 1, and that's the one from rat 2. Can we see why the behavior differs? That's a very different question. Then you really have to go into specific neocortical columns and details therein. Yes?
What do you exactly mean by difference between whole brain simulation and whole brain emulation?
Oh, well, I didn't touch that one, but I generally just move from using the word simulation to using the word emulation when we try to get the predictive behavior right within a certain error term. So the idea is if you're doing a simulation, then you're trying to get the general function, learn something about the general function, and it can be qualitatively right, but not applied to any particular individual, for example. So let's say it's not useful as a neuroprosthetic, but it is useful as a learning tool. And in an emulation, I would say it's the reverse. You're really interested in something specific, something that could be used as a neuroprosthetic, for instance. So it's a matter of how many parameters you fit. And somewhere you make this slide from simulation to emulation, and it is a bit vague, I agree. Okay, unless there are any more questions about this particular bit of research, then I'll go on. What is this? I see some movement in the top booth there, but I'm not sure. Oh, it's gone. Okay. Oh, that's okay. Is that for a while? Okay. Then another one. This was an interesting recent controversy. Dharmendra Mantra. He was in the news because he got this DARPA grant, the Synapse grant, and is really also looking at very large-scale systems. He wants to do cognitive computing, but he comes from a computer science background rather than a neuroscience background, so his approach is quite different, and it's more aligned with some of what we've seen here so far and what the AGI community may be comfortable with, which is to use some general principles and try to arrange a system large enough and competent enough to carry out the kind of computations you would see in the cortex. But at one point he said, well, from the general connections we have and the kind of flow of data we see and the number of neurons we've got, we've basically reached the level of simulating a cat brain. And this set off Henry Markram, who immediately responded with an angry letter saying this is absolutely untrue, how can you say anything like that? And it went back and forth for a little while until now it's becoming clear that what Dharmendra was trying to say is just, well, we've reached the ability, the hardware and software capability to simulate something powerful enough to be at the level of what you would need in order to compute the cognitive functions in a cat brain. So,
Markram called it a hoax.
well, yes, he called it a hoax, yes, I know. I don't want to start repeating all these things because I think that this is all going to be online, and so I don't want to attack anyone personally. I'm just saying there was a controversy, and now it's becoming more clear what everybody means. And it is an impressive feat. I mean, do you see here just the 147,000 processors and 144 terabytes? Okay, maybe in 5 years people will poo-poo that and say that's no big feat, but now it does look like one. That's kind of, it's cool. Then we've got something, again, from another angle. Itzikevich, well known for his work, having developed a very nice function for neurons that can do pretty much what integrated fire neurons will do, but, and in fact, in some ways better, because they can simulate the response functions or response curves of a very large range of neurons very well, but without going into all the details about channel conductances, etc., that you see in the Hodgkin and Huxley models that are used in the neuron simulations that, for example, Markram is using. And these neuron models that Itzikevich uses, they enable him to make very large-scale simulations that run quite fast and with a great amount of detail. And what he's done is he's been working on large-scale models with microcircuitry. This is just a little excerpt of one of those microcircuits. And he has so far been able to simulate about 100 billion neurons simultaneously, so that's human brain level, with a quadrillion synapses, and has observed alpha and gamma rhythms in there and clusters of moving activity. He used 27 processors, processing for 50 days to achieve 1 second of simulation. Now I'm guessing that if you port this to the architecture that Dharmendra was using, then you get a much better result, but in terms of time. But still, let's talk a bit about what was actually simulated here. Itzikevich, at least in his initial models, did not store any of the synaptic weights. So at every instance, every time that the model was going through another iteration, all the connections would be reinstantiated, and then it would be run again. So this way, you save yourself a lot of trouble. You're not doing the real learning in the brain. Instead, you're creating something that has the right kind of connectivity and the right number of elements in order to observe these large-scale phenomena, such as the alpha and gamma rhythms and moving clusters of activity. So what he's done is he's made a really big simulation, but it's not something you could use to, for instance, make a neuroprosthetic, just to go there. And yeah, he's interested in looking at multiple scales, which is also a hot topic right now. So combining things from single neuron simulations, microcircuitry simulations, and the connectivity throughout the brain, which is basically the white matter, and using diffusion tensor imaging to extract that kind of connectivity to a great amount of detail. Okay, and then we have, it's the other side of the coin. All of what you've seen so far was really simulating activity, looking at a function and trying to do that at large scale and high resolution. This is anatomy, structure. One other way to try to understand what's going on in the brain, if you're going for whole brain emulation, is to look at the underlying structure and say, that is my basis of information. I'm grounding everything in the detailed, fine structure of the brain. And then if I need to know what the activity is going to be, well, if I know enough about the neurons, if I know enough about the synapses and about the chemistry, then I can reconstitute that activity by inferring from the structure of the neurons, from their morphology, what type of neuron it is and how it would behave. So you look at the structure first. This is also called high throughput microscopy. So the idea is you embed a brain in plastic or you freeze it or something, and then slice it really thin, and all those slices are imaged, and then you put all the image stacks back together, you extract from that information about where your significant components are, so for example, neurons, dendrites, and axons, and if you're interested in them, glia, and whatever else, and then try to create simulations with it. There's been a good deal of work on the acquisition side, so on this, how do we get these stacks of image data at different levels. Yoon-Suk Cho, working in Texas, he's been working at the light microscope level. So, and this is in fact what I'm going to try to run right here. This is a simulation of the stacks having been put together again after, well, a small piece anyway, having been put together again. Now, how do I start that up? Let's see. Let's see if we can get this running. I don't seem to see it. Let's, oh, there's something there. I'm not sure what I'm having this problem. Okay. Right, so this is basically a reconstruction of some portion of rat brain, which was run through their system called the knife-edge scanning microscope, where they extract slices and then from that image with light microscopy. I won't go into the details of how it happens, although it's really fascinating. And then you get these kinds of stacks of images. So it's not that important to our current conversation, but just to know that that's another way to go about things. Then you have, let's see if we can get this back onto full screen view again. Then you have the Lichtman lab at Harvard and Hayworth. Together they've built something called the Applin, automated tape collecting lathe ultramicrotome. It's more detailed. It allows you to collect a library of these slices of brain pieces so that you can then examine them with electron microscopy, for example, to get an even higher resolution so that you can look at the details of vesicles inside the synaptic terminals, things like that. And there's Denk, who's been doing his work here in Europe, basically ablating the surface of the brain and making images of that as it goes along. Yes?
Just to check, this slice scale, human and right, seems very equal, right or not?
Sorry?
At this slice scale, human brain or rat brain are roughly equal. Is it true or not?
Oh, when you're looking into the system like that, to do this circuitry, and you just see the tangle of dendrites and axons, yes, there's no way to distinguish whether you're looking at a rat or a human brain, because we can't see the larger anatomy here. Okay, so this is the other approach. There's a reason why that is complicated, and one of them is, of course, that everything you say about function is inferred from structure, and there are many ways you can go wrong with that. Okay, then I have to bring myself into this, of course, even though my results are much, much less impressive. My part of this is really on multi-scale integration also, and that's really the emphasis of my lab right now, is how do we integrate different scales of activity in the brain? How do we integrate from the level of the system, so system models, with the level of individual neurons spiking, and what do we need to know about the levels below that? So it's an investigation of the data levels. And just to show you a movie as well, because I have to do that all the time, here we have just some development of neurons with okay, I better get out of this again. Hello. This is being difficult. Yesterday it was all working wonderfully, but that's the miracle of movies. Okay. Let's go back to the start first. So what you're seeing, this is based on a platform that I developed called NetMorph, in which you can develop neurons from their earliest state out to X number of days and see networks develop with axons and dendrites with a statistically correct morphology. So again, this is a construct, this is based on statistics, and is again used to extract general principles to look at when you get to day number 7 and you have the kind of connectivity that you see at day number 7, can we already observe oscillations that resemble some oscillations you find in the brain? Things like that. Or do we find that the number of connections at this distance from a neuron are approximately that many, and how does that relate to what we see in the real brain? So what can we discover about connectivity that is already an emergent property of intrinsic outgrowth and development details in the neurophysiology and neuroanatomy of the brain? So this is still a far cry from anything related to neuroprostheses, but it's a lot of fun to do. Just switch back to full screen. I hope that was the last movie. Okay. Right. A few big questions are coming up. First question, how do we describe what we're looking for when we're thinking about scope and resolution? Well, first, let's try to define what's going on in a mind. And I would like to break it up into 3 pieces, first of all. And I really hope that after describing this way of looking at it, that I can get a lot of feedback and comments about that. Something I call mind state. Okay, it's a snapshot of mental activity. This is what elicits awareness, elicits external behavior. Of course, you can say to have awareness and behavior, it needs to be more than a snapshot. It needs to be the dynamics. But what I'm saying is whatever happens, if you take a snapshot of what's going on in your brain right now, that's state. Then, first order quantification, mind transition functions. Describe the dynamic development of mind state. If you are given a state at time t, what does it take to predict the state at time t plus delta t, or dt in this case, with some acceptable variation and, well, in that restricted time span. We don't want to make delta t too large. But if we have functions that can describe this transition between one state and the next, very useful. So that's my next point, is first order quantification. And then second order quantification. The brain learns, it changes, modifies. These are update functions. These are functions that update the transitions between states. So again, they are affected by mental activity as well as sensory input, etc. It modifies parameters, it modifies structure. And you could say that if you look at all these changes over time, it's a history of mind state. It's a memory of mind state. It's basically a memory of our memories. Now, what is the minimum data that retains the information theoretic mind? So when we think about mind state, mind transition functions, in a second, yes, and mind update functions, what is the lowest level of data we need to go to? Or what is the resolution we need to achieve to say something about that? Okay, before I go into the possibilities, let me just see what the question was. Yes?
Do you make a difference between mind state and brain state?
Not really. The thing is that when I talk about mind, I do so on purpose to try to not be distracted by the details of the substrate that it's running on, because what we're trying to do anyway is a whole brain emulation. So we're trying to move away from this substrate and say, what can we say about the way it works? What are the functions and mechanisms? And so when I'm talking about mind state, I want to know what describes the state of the functions, what describes the state of whatever's happening to the information there. Not so much the state of a particular vesicle in a terminal of a synapse or something like that, because we're not interested in that. We're interested in the information in the brain. So that kind of state. But it's good that you say that, because of course to find out what the mind state is, you need to look at all this mechanistic stuff, and that's what's on this slide here. Is it enough if we look at the activity in some big pieces of the brain, voxels basically? And this could be, for instance, what you look at in MRI. You look at the uptake of oxygen, you look at the flow of water, and in some cases now you can also look at the flow of dopamine. That might be enough to say some things about the brain, but I think most people agree that's not high enough resolution. The activity of groups of neurons. Well, we often say that in neural networks, it's not so important what the individual neuron is doing, it's more important what the network is doing. You can say they're robust, you can shoot out a few of those neurons and the system will still work, more or less. So maybe we just need to look at groups of neurons and get some idea of what the activity of a group is, small enough. Then, time-averaged rate activity of individual neurons. Well, do we have to look at what every neuron is doing and how active it is basically within 1 second or 10 seconds or something like that? Or do we need to know the spiking activity of individual neurons? So do we need to know when each particular neuron is firing? Do we need to know this about the entire collection of neurons in our system in order to make that prediction from t to t plus delta t? Do we need to know what the membrane response is in each of these individual neurons? So do we need to go further than the spiking activity? Do we need to describe the function of that activity? Or do we even need to know within the morphology of the neurons, in their dendrites, in their axons, at individual spines where they connect to other synapses, what is going on there at every moment in time? Do we need to know the distribution of memory potential there, which is basically another way of looking at calcium distributions? Or do we even need to look lower, look at the molecular level, the transition of molecules, and the balance that exists there, intracellularly, extracellularly, through the channels, etc. Do we need to know the flux and all this other stuff? So where do you have to go if you want to make real emulation? This is a major question. Yes?
Yeah, well, my suggestion would be to stop at the... my suggestion would be for the first order emulation to work, to stop at the spiking activated level. But for modeling the update function, you might have to go lower, because we're not exactly sure how that happens.
It's really interesting that you say that, because this corresponds with my unofficial survey of what most professionals think about this when I pose the question. And it's that, on the whole, the majority seems to think that for the mind state, saying something about spiking activity is enough. If you want to say something about transition functions, okay, maybe you could still make do with spiking, but you might have to go a little bit lower. Not so sure, that the opinions differ. And then update functions, there's at least, almost everyone thinks you have to go below the level of spiking and know more about what's going on in, say, for example, this part of the neuron where something is being elevated and strengthened, and when you get LTP, etc. So there is that.
Yeah, and I have a very specific suggestion for testing whether the spiking activity is correct. I think BLX work should be taken into account. So in the information coding, efficiency should be measured somehow. I mean, that's your magic. I don't know how you do that, but it should be done, I think. Thank you.
Okay, but I'm mostly just posing a question here because it's still so unanswered.
What was your intuition? You said that you see as a result of your informal surveys, but where do you think you feel?
I'm sorry, I missed most of this because of the sound of shoes.
Do you agree with standard beliefs in respect to that?
So with standard beliefs?
You said just before that you think when you
Oh,
survey
if
people.
I agree with that?
Yeah.
Well, yes, it was also my initial assumption, so nothing different there. Yeah. Okay. Now, one thing that we can say about all these different levels when we look at the resolutions that I just went through is that each time function and structure both seem to be important. So that's something we can take along. At each candidate resolution, this is the case. We need to look at the characteristic parameters of the dynamic physiological functions at that level, and we need to look at the structure that governs the interactions between the components. That's not so surprising because we're talking about neural nets. But an interesting thing about this is that there may be differences in what level you have to go to in terms of structure and what level you have to go to in terms of physiological function. So you may not have to go to the same resolution for each. Also, you can sometimes use information about the one to infer the other. We have, for example, the case where if you know a lot about the activity of the individual neurons in an array of neurons, then you can extract the functional connectivity to a large degree, so you don't need to look at it with a microscope. You can find out what that connectivity has to be, at least within the realm of that activity that you saw in your experiment. There's a bit of a problem there that if that's only a very short time period, that you may not have explored all the possible forms of activity that could be there, so you may still need to look at the actual structure. And conversely, if you have the structure, well, that's what I was just saying before about these microscope methods, you can try to infer something about function from that. So the two, structure and function, it's interesting that they're both there, and it can be a problem, but it can also help. Now, when you're looking at what are the limitations, why would we stop at a certain resolution, an important thing to consider is what, in a particular region of the brain, are the limitations of the functions that are being carried out, the operations being carried out in those regions. Well, there may be a certain resolution at which, if you go below that resolution, if you go to an even higher resolution, and I shouldn't say below, but above, in fact, that you don't get any extra significant information that's useful for these operations. So what are operational constraints, for example? Intrinsic noise is one of the possible operative constraints. Tolerance to parameter variation, that in fact you have a robust system that depends on many neurons. You see this, for instance, in motor cortex, where really a lot of neurons seem to cooperate in a very vector-oriented way to drive motion. Requirements for robust performance. Okay, I kind of went into that already, so I have to say that again. Now, how do we discover operational constraints? One thing to do is the modeling studies. We've looked at a few of these possibilities already, to then explore the parameter space in there. But it's very important, again, this whole concept of grounding, that we have to go back to experimental measurements that are done in vivo. It's really the only way to know for sure, because models allow us to do so many things. Models are wonderful playthings. You can make things with them, and that's different than analyzing things. Now, okay, why do I mention this here? Okay, yeah. Why in vivo? Let's consider the post-mortem approach for a moment. And at the top, I just once again remind us of the neuroprosthetic direction that this can take, and that the work that both Markham are doing and that Berger are doing, both of these, they can be useful for that. But if we look at a post-mortem extraction, this large-scale, high-resolution imaging, then we rely entirely on inference for our function. And so the quality and applicability of the inferences determines the performance of the emulation. Why do we need to combine large scale and high resolution? If we look at just low resolution combined with large scale, such as an fMRI, there are a number of problems. The experiment you're carrying out is usually limited in duration. That means you're only exposing a few of the functional connections. Only whatever's being used in that task at that time is providing some difference between the images so that you can say something about activity that is specific to that task. Also, this allows you to make limited inference about the underlying complexity that's really there. The underlying complexity, what is in the structure and in the learned synapses in the brain, would allow many more types of activity than you see during such a study. High resolution and small scale data, well, that's, for example, when you're doing multi-electrode recordings. Again, you only look at a limited set of interactions, and you're really just picking one spot and you're saying, oh, while all this is going on in the entire brain because the rat is running through this maze, those few neurons are doing this. That doesn't tell you very much about how a modular system works, something that has really billions of neurons. It's like I'm saying, the comparisons as if you're trying to understand what a painting is by looking at a really small snippet, a tiny pixel here and a pixel over there. And then maybe you can say something about the style of the artist, which is like saying something about the functions, the underlying fundamental functions in the neural system, but you really don't understand the picture. So lack of knowledge about structure restricts knowledge of behavior. Well, you can't extrapolate unseen behaviors in any of these experiments. What aims do we have for in vivo measurement? We want to combine structure and function information and thereby improve the probability that we can also predict something for those episodes that we didn't see. And in a sense, this is similar to the challenge that the INCF recently held for the single neuron computation, which is, okay, how well can we simulate what this neuron is doing? Can we simulate so well that we can predict all the spike times? And then you have a set of sample data, and you want to be able to predict those spike times, but with the model you've created, you also want to be able to predict the test set, those spikes that you didn't get when you were building your model, and you still need to have the right result. But now, up a few levels into the whole brain eventually. We can ground many structure and function parameters through direct measurements of morphology and functional correlates. We reduce the free parameters, and so we don't need to infer as much from our recorded data. And there's this whole big topic about what kind of deviations are acceptable. So we have this t plus delta t, and we want to be able to predict what's going on there. What is acceptable? Very big topic. I'm not going to go into it at all. Okay, some measurement requirements. And I don't know what my time is right now, but I know that I may have to hurry up. I'm not sure. How far are we in? Does anybody know how much more? Sorry?
10 minutes more. You have 20 more minutes.
Okay, so, yeah. Well, I'll have to hurry. Okay. We'll want to do episodic recording in vivo, make physiological measurements and morphological measurements. We would like our recordings to be distributed over large scales, so throughout the entire brain, into a large number of recording sites, have many, many recording sites, preferably all the neurons in the brain, maybe all the synapses in the brain, some number that large. And high resolution at high rates. So we don't need high resolution just in space, but also in time, high enough at least to capture whatever's going on. For computer scientists, that may not be a very high rate because things are pretty slow in the brain, but it's high enough when you think about the bandwidth requirements to get this kind of information out of the brain. Now, just a very quick list of some existing imaging technologies. I'm not going to discuss them, but just show them to you, and it's not even a complete list, but this stuff is all there, and it all suffers from exactly the kind of problems that I brought up before, low resolution, and another problem, not being able to see the things you want to see. You may see oxygen uptake here, but that's not necessarily telling you what's really going on in the system. A lot of the things we would like to see don't produce signals that are easy to capture with imaging technology. Even though imaging technology has some really nice features, it's not so invasive, and it can look at the whole brain. So there's a reason to like imaging technology. Now, existing in vivo interfacing, well, You would like to be able to do chronic implants because you want to be able to follow the activity over a long period of time so that you're not stuck with this problem of looking at only a very small time window and having to infer what other kinds of complex activity could be going on. Chronic implants are problematic. A lot goes wrong, and so really we don't have any good chronic implants right now, even though we need them even just for stimulating electrodes, say in deep brain stimulation, so that we have implants that can last for a long time and to give people with Parkinson's disease relief. So there's a good reason to develop that, and the medical industry is pushing that as well. Recording through those interfaces, multi-electrode arrays, it's restricted to a very small sample of the brain network. It fails to sustain recording from the same cells over long durations. And again, we have this problem that it may not be showing us what we want to know, because now we're just measuring the electric currents, and it may not be telling us enough about other things like calcium deposits that are in these buffers inside certain areas of the neural morphology, or may not be telling us about diffuse chemical transport and about temperature. Temperature also has an effect on neurons, and that's largely unexplored. So what are the aims for good recording interfaces and imaging techniques? Let's just list them. There are safety requirements. To make this useful, to make it something that you can actually experiment with, especially with human subjects, or that you would be able to use in a neuroprosthetic, you want it to be harmless, so it's not going to cause some major disruption of how your brain works. Low impact on activities, so for instance, just having a long cable that's dragging out of your head, if you have an implant there, that would not be very useful, especially when you're trying to go skiing or something. Then safety of the interface implantation or functional scanning procedure. The procedure used to put it in can be dangerous. And the procedure of using it, so remaking the recordings, can also be dangerous. Let's say if you have to use high intensity x-rays or something like that. Where possible, avoid major surgery. Anytime you're putting a port in someone's brain, that's major surgery. It comes with all the risks of major surgery. You want to avoid damage to healthy tissue. Then performance requirements. We want a reliable interface or scanning technology, something that will work for a long period of time and that will give us reliable results. We want large-scale neural interfacing or recording. Interfaces should be distributed throughout the brain. We want brain-wide scans in the case of brain imaging, a high rate of data acquisition, or high enough. At least it's not that huge of a problem in terms of the acquisition, more of the bandwidth for transmission of the information. High spatial or component resolution during data acquisition. That's, of course, the main problem that we're talking about. Precise, reliable, absolute, or relative location registration. So if you have something that can tell you about what's going on in a bunch of neurons, but it can't tell you where they are, so how these measurements are related to one another in space or in some kind of connectivity, then it still leaves you with a big problem. So we need to have some registration of location. Now there are a number of different directions that are being explored, and many are beginning to point into a common direction, to have sort of a common theme. On the aggregation side, when we want to aggregate this whole brain data that's coming out of whatever you're using to measure information, you want something that's non-invasive, and so imaging is a good way to aggregate data. So one way to do this is to use imaging technology that's at large scale, it doesn't harm people very much, but you need to make the information you actually want to see visible, because you cannot see most things with imaging technology alone. So the imaging technology then becomes just the aggregator of data. The detection is often aided by putting something in the brain. Now, I'm not talking about microelectrodes, I'm talking about things like dyes. And this has been used for the last 10 years. We've been putting dyes in order to improve what the imaging technology can see, to make things visible. Sometimes it's even useful for treatments. I won't go into detail on that either. And most recently, so I'm just going to hurry up a bit more. Oh, sorry. Most recently, here we've got quantum dots. It's beginning to replace the organic dyes. A little bit more about quantum dots. They're really tiny semiconductor pieces, crystals really, that have conductances that are determined by their size and shape. So these have special properties in that sense. They were developed by Bell Labs. They can be functionalized. So Q dots can bind to specific sites. It's useful if you want to explore tumors or synapses, which would be, in the case of whole brain emulation, that would be very interesting. You can use them for extended durations. So they're very stable. They don't break down easily. They tend to be resistant to wear and tear from multiple imaging. And they can migrate with individual cells. So you can also watch the flow or the transitions where cells are moving from one place to another. There are, though, safety concerns. These have not been fully tested in sense of toxicity. So quantum dots are not yet in use or applied to human imaging. Because even though we can coat them with polymers to make them not immediately toxic, they can release their toxicity if there's UV radiation, for example, but in the absence thereof, in the polymers, they seem relatively safe. But we don't know as much yet about how these quantum dots, once they're inside your system, how do they actually disappear? How are they dissipated, excreted? What is the process there? It's not so clear yet. This image here is just an image of a couple of neurons, and I think this is actually from a rat imaged with quantum dot. With a quantum dot technique. Okay, now just to sketch out a general approach, not specifically with quantum dots, but this idea of putting something in the brain to improve what we can see and then using imaging to get something, to aggregate the information. We can enhance what we want to see in there by introducing nanoscopic agents. I'll just say agents because there are a number of different ways you can go with that. And what you want to do is you want these agents to be tailored to work together with the imaging technology. Normally, our imaging technology is restricted to whatever signals we can get out of the brain. If you have an agent inside the brain, you have the capacity to engineer the two to work together. So then you can turn otherwise invisible information into visible information, bring it out. And this is just showing two different approaches. You could have this nanoscopic agent inside the brain coming in, for example, through the vasculature. You just get it injected into your blood, and wherever it is, it receives some power through some radiation that you would supply, and then it activates, produces some kind of response, which is detected. The other concept is more of a passive concept, where you have, this is like the dyes, that you've got something that changes properties when there is activity going on, there's a biological signal it's trying to detect, and that changes its properties when imaged. So you've got two different ways to go about it, an active approach and a passive approach. Now, some requirements for these agents. They have to register their location in some way, or we register it for them. If you're using an imaging scan and you can see where these agents are, then you're already registering their location. But it may be possible for the agents to self-report. You could have a way to create some communication chain that brings out the information of where they are, at least in a relative way. I'm next to this agent, and next to that one, and next to this one. You could have multiple specialized agents that achieve a desired result cooperatively. This is not a bizarre concept. This is just exactly what goes on inside our bodies already. We have a large number of different types of cells that do very specific things, and they work together. They do things together. That's how our body operates. So you can do the same sort of thing. You can have agents that cooperate. One is specialized at obtaining power, perhaps the same way that our cells obtain power. Location registration, detecting the biological information that you're interested in, whether it's temperature, potential, chemical, something like that, and convert it into a scannable response. Now, there are different kinds of agents we could be thinking about. One are engineered devices. So the idea is to make something at nanoscale. You could produce it perhaps with the same type of technology used to produce to work in the semiconductor industry, or these devices might produce themselves, replicating just the basic molecular nanotechnology approach that is still wonderful future music, but not quite there yet. Another approach is engineered biology, which seems quite promising right now. What you see down here is an example of an engineered bacterium, synthesized and assembled. This is in the Venture Lab, and also in George Church's lab at Harvard. They're working now on, I shouldn't say too much because this isn't really going on yet, but they've started working on approaches of engineering cells to help us obtain information from the brain. They have a new postdoc exam, they're working on this, so this is still, it's coming up. And the idea is that instead of building silicon systems or some other artificial system like that, we can build with the same blocks, with the same building blocks that are already being used in our bodies, but then engineer them to do certain tasks to help us turn information into something visible, the information we want to receive. Okay, so what do you do with any of these kinds of agents? Probably a good way to go about it is to inject them into the vasculature. The vasculature supplies necessary nutrients to each part of our brain. It goes right up to every neuron, so you can reach all the neurons by going there. And if you have something that's already as small as the cells that are traveling in the vasculature anyway, then you can do this. You can either allow the distribution to happen naturally, or you can try to target the distribution by using chemical signals, for example. You can use, again, inspiration from the brain and how the processes going on inside our brain use targeting and manage to get to specific locations. You want to record at very many sites, so you need quite a lot of this stuff. Better not be dangerous to us. Again, the toxicity issues and other interference measures that need to be taken care of. And you need the responses to come out to external storage or to be handled by some type of delivery agent. So you can image the data, you can maybe pass it out some other way from one agent to another. And, well, if you still need additional information with a fine structure, then you might go to one of the other approaches I discussed earlier, such as the post-mortem approaches to get fine structure out of it. But it's a little difficult to consider right now because we don't have details about the in vivo procedure. Now, just a few final bits and pieces. Another interesting way to go, brain repair by nano-engineered neurons. Instead of just looking at what can we put in the brain, you can say, let's take some pieces from the brain, like neurons, and see if we can learn about them and learn about the fundamental properties by putting them on a substrate where it's easier to observe all the details of what's going on. If you put them on a carbon nanotube substrate, then you have a very good conductance substrate that has an affinity, or that the neurons have an affinity towards. So this can help you, perhaps, learn more about how these systems grow, what they do, and how they act. But it's still unclear how neuronal networks react to adherence to conductive material, so this is an ongoing area of research. And similarly, nanowires. Sometimes, in a very futuristic prospect, nanowires have been considered as a way that you might get information out of the brain by basically putting some sort of mesh into the brain, but that's not what I'm really talking about here. It's more about how could you use nanowires as a substrate on which you can then implement either a neuroprosthetic transplant or which you could use to map neural circuitry. And then my final remarks, okay, just try to recap a little bit. The basic problem I wanted to address, two of them. One is the data levels. So what's generalizable to all brains, what is more subject specific, and how can we integrate across multiple different scales? What are those scales anyway? And can we use inference? Then the other basic problem, well, if we need to explore at high resolution and large scale, then what can we use for that? One approach is the material science approach, and my lab's involved in that to some degree because we have a collaboration with a part of Technalia that does this kind of material science with carbon nanotubes and all that. And so we would like to explore that approach, but personally I'm very attracted right now by the cell biology approach, and I hope that I'll be able to collaborate with some interesting projects there. And then, of course, you need the imaging technology to connect well with these developments. And finally, funding. Always welcome. It's very necessary, and yeah, that's going to move things along nicely. And I think that's it. I just have a couple of references. So if there are any final questions, please.
So, question regarding your example of the painting. So, just to clarify, if you get to see a whole painting, because we have a nice visual cortex, you can actually understand it. So you sort of implied that if you could see the whole brain at a high resolution, that you would understand it effortlessly in the same way that we can effortlessly understand a painting, which seems a bit of a stretch. I was wondering if you could clarify what you meant by that.
Okay, I did not mean to say that a painting is a good analogy for whole brain emulation. If I said that, then I have to backtrack a little bit. It was really an analogy that was just aimed at trying to elucidate the problem of working with these individual recording sites. Whole brain emulation goes beyond that because yes, you need to know what's going on at the detailed level, and yes, you need to know how it all fits together. But then running an emulation like that is a separate problem, and understanding what's going on in it is a completely separate problem. It's another, it's another dimension that needs to be investigated. But emulation is at the same time an opportunity to investigate that while you're working on it. We were talking about that in the case of Markram's work. But it's also a tool where when you have some emulated examples, be it a Drosophila or be it human, then you can use that to do this exploration and try to understand more about what's going on in it. So it's not quite the same as saying it is a painting.
If I could ask just a related follow-up. So Ray Kurzweil claims that brain scanning technology is improving exponentially. So I was wondering, as a practitioner, whether you agree or disagree with that, and also whether the relevant dimensions along which it might or might not be improving exponentially are the ones that actually matter to you in terms of building whole brain.
What about just 2029 timescale for having a human upload?
Yeah. Well, okay. I'll try to answer both questions. Let's see. We'll start first with the exponential development in imaging technology. I hope and wish it's true. I don't know, because the problem with these exponentials is that it's often kind of retrospective. We look at something and how it's developed, and we say, oh, look, yeah, look, this fits an exponential. So wonderful. But that doesn't necessarily say that what we're interested in is going to develop like an exponential, because driving a field of research to where you need it to be is a different problem than just looking at what happens to be developing. And in the imaging area, I think that, yes, we see increases, important increases where, for instance, computer power is important, so that the analysis of the images gets better, or so that you can capture more data more rapidly. You can have more detectors at the same time, and things like that, that's working very well. But that doesn't really change the nature of the imaging technology used. It's not a breakthrough in how we can see the type of information we'd like to get out of the brain. And I think that really different approaches, such as what George Church and Venter and there may make possible, those are really the ones that can change the equation. They can give us the information previously not available to us. So that's just my take on it. But I'm trying to follow this in many ways. And one of my main tasks really is to try to pull together all these different possibilities and keep tabs on what's going on, to try to steer it in that direction. And I would love to create an exponential like that or be part of it. And if you can, it's like what Rod Brooks says, when you see exponentials, you should jump on board. You should try to get on the bandwagon. And that's true. If you see an exponential you can use, go with it. Yeah. And now that other question, 2029, if we have the right exponentials, yes. Okay.
So sort of related to that last question, there have been a lot of recent advances in imaging technology in terms of, for example, combining fMRI with MEG at the same time, so you get some nice spatial and temporal resolution. You've also analysis techniques like using support vector machines to analyze the data, so you can actually get to sub-voxel features by, people have used it to look at orientation columns in human visual cortex, which are below the resolution of a single voxel. But if you combine all of the activity from many voxels, you can still infer something. So, and then you also did mention like lesion studies as well. So to investigate causality, where using TMS to cause lesions, temporary lesions. It seems like you need all of these approaches. I mean, maybe you would agree with this. I mean, to answer the questions, depending on the exact question, different approach might be more suitable to the, and probably you need all of them together to answer these bigger questions.
Okay, I think that for the fundamental research, to try to discover what is the level of data we need to look at and what are the functions, yes, a combination of all approaches, every piece of study has value. I think that lesion studies are valuable. I think that combinations of imaging techniques are valuable. But if you're trying eventually to do a whole brain emulation that is subject specific, so you want to be able to capture in vivo or post-mortem enough to be able to create this neuroprosthetic of an individual animal or an individual person, then you cannot just take all these different techniques and kind of put something together. It has to be something that will provide the information you're interested in, and that doesn't necessarily change by combining MEG and fMRI or something like that. It doesn't necessarily give you new kinds of information. It doesn't tell you anything about the calcium uptake in the synapses or something like that. Although, yes, I know there's that whole dopamine.
Also, it may give you
yeah.
good targets for the single cell recording and give good information that you can then go and investigate in more detail.
Yeah, that's why imaging is always going to have its uses. There's no doubt about it. In fact, this whole other approach that I'm talking about uses the imaging then as an aggregator as well as the way to get information out. So I'm not against imaging in any way whatsoever. I think it's very useful. It's just that I think we also need to look at these new approaches to get at the information at the scale, at the resolution, and with the types of information we're looking for.
As far as I can understand, today there is a huge debate in cognitive neuroscience about the usefulness of medical imaging for cognitive purposes. You may know the famous paper of Pascher on voodoo correlations among fMRI images and cognitive aspects of. So I understand that your work is very useful for medical purposes, but I would like to know your position about the usefulness of understanding the mind, as you said before. Thank you.
Yeah. Well, what you're saying, though, refers specifically to the problem of the imaging technology that we have right now, which is that when you use MRI, for instance, you're really only correlating differences and only about certain kinds of activity that you can observe. And so then you have a certain amount of information there, and you start to make all your hypotheses and all your theories depend on that. You say, this is my information, this is what I base it on, and yeah, that can be completely wrong. So I agree that that's a problem, and I think this is sort of tying into the same issues I was discussing in the talk today.
Well, I really much liked your second definition of the problem, the formal one, the behavioral one, which is not so. And my question was about, well, the typical ethical issue. What do you expect that this approximation is good enough to start thinking about, I have to stop because I have an ethical problem here? So can you comment on that?
Implications, okay. Yeah, then you're opening up a whole new topic. Okay, we could talk about this for a long time, and I see Anders is already trying to ask a question there, and he's exactly the right person to talk about this. Because of course, it's just like an AGI. Once you start to create something complex enough and sophisticated enough, and we're talking about techniques here that are going to be, I guess, at least highly invasive in the sense of obtaining a lot of information about you, first of all, and then doing something within the information and then creating something that would again act like an animal or a person does. So, okay, we have ethical issues galore there. And just like you have in AGI, I think that you need to approach those one by one for each specific case. You need to think about what is to the benefit and what is not to the benefit of the person or society involved. And I think that that does not take away from the general field as having value, both in terms of allowing us to understand more about the brain, or to engineer for the brain, or to help in medical situations. So it doesn't take away from the value of the field, but it does mean that, like we've discovered in so many areas now, you can't just waltz into there and do anything you want to without considering the ethical implications one by one. Now, I've forgotten some of the specifics of your question just there, so maybe you want to, if I haven't quite answered it, please.
Perhaps with your the progress of this research, you create some kind of Frankenstein thing, and you, what we have here, and perhaps I don't know, maybe you don't know whether you have to disconnect the system or keep it living. Well, these kind of issues.
Okay, I agree, but this is, we should all have this discussion because the same thing applies in AGI. Once you create a system that you think is intelligent and it's active, then do you have the right to turn it off? Yeah, it's a big question. And maybe Anders wants to give me an answer.
I'm actually trying to write a paper about this. Many questions. Thank you. I'm actually working on a paper about this right now, and many of the questions are essentially basic medical ethics questions, basic research ethical questions. But then they get this added spice that we probably won't agree, at least early on. For example, does a simulated lab rat have an experiential content? So if I pinch its simulated tail, is it actually going to suffer? And does that mean we should have software rights activists complaining that we're treating our simulated animals wrongly? That's going to cause a lot of interesting confusion and going to keep us bioethicists probably in the battle for a long time. But the really deep problems, of course, start when you get into the level where you reach human intelligence. I think in general, most ethical systems end up with saying that, yes, intelligent systems should be treated with respect. The problem is, of course, what respect and intelligence means in this context. And then, of course, there are potential very, very big social issues later on. For example, Robin Hanson's economical model suggests that there could be some very scary implications here, just like for AGI. But I'm going to talk a bit about that actually in my own workshop presentation. But the short answer is yes, there are various interesting and Frankensteinian possibilities. We need to be on the lookout for that. But as long as we're in the lab, we're actually having a lot of lab roots. That's not the problem. The real problem starts when we get something that's very different from the normal lab research, because then our intuitions might not be the right ones to guide us. And also our regulations might also say nothing. So personally, I don't think we are allowed to pinch simulated or emulated rats. But right now, the rules of good treatment of software don't exist.
Well, that sounds like it's going to be a very interesting talk that I'd like to see.
From the standpoint of AI and computation, I'm a little confused. So if I put it pointedly, using terms like hoax, it seems to me the whole thing is a hoax. And it's just massively ironic that Mark could make a statement like that. Okay, here's the problem. I'd like to get your reaction. We have a bunch of theorems, okay, that tell us that nothing could be more stupid from the standpoint of computer science when trying to figure out how to compute a function, to go and get a piece of hardware that is computing that function. Okay? Go get a supercomputer. You don't know what it's doing, but you're going to check it out and figure out what it's doing. It's not possible. Okay. Now, can you or anyone else give me a single example of a function, any kind of function that would be of interest to a computer scientist, which we now know how to compute because we found the device or sub-device instantiated in the brain that shows us how to compute. We don't know how we generate sentences. Right? We don't even know how we do basic arithmetic. But we're going to simulate entire brains and provide insights for a field like AI or AGI. It's so preposterous that it takes the human race to make this a reality. Now, medically, absolutely. Maybe you can figure out, why this tumor I have, if I have one, God forbid, can be taken care of, or how I can be augmented. But that's different. We give people drugs to help them with depression. We don't understand depression. It's just the way it works. We take a crap shot. they feel a little better, that's how medicine works. But that's not how rigorous AI and computer science works. So help me understand. Maybe the answer is there is no connection, or there is no doctrinaire assertion about a connection, but help me understand how this is relevant at all. Because I just don't get it. And again, the Blue Brain Project,
Right. Okay, so I'm glad that we don't have to talk about the medical part anymore because that's clear. It's true that there hasn't been a lot of effort to clearly delineate what kind of benefit we've gotten from looking at the human brain or looking at details of biological function in order to learn something for computation. Even though if we take the larger picture and we look at, for example, we've looked at how genetics works and we've learned something about, how you use genetic algorithms and use a similar system there. Okay, something from biology. Also, we can say as an example, we've looked at how neural networks work and we've learned something about liquid state machines from that and we can use that to do interesting things. So you could say we've learned something about potentially interesting tools or utilities that come from a kind of computation that we can observe carried out in the brain. But it's true that is different than saying we can take a whole brain emulation and learn something about the brain from that. Still though, I think that there are a lot of things about the way that the human brain functions that haven't been taken into consideration in AI because we don't know enough about them. And I just had a discussion yesterday about one of these examples, and it's interesting. For example, there's different cycles, different phases that our brain goes through. One of them being sleep cycles, and why do we have sleep, and what goes on during sleep? How does memory consolidation happen? Things like that. Why is it modular? Why do these things happen in phases like this? First then, and then. Does it somehow have something to do with filtering the data that's coming in an online runtime system that's receiving new experiences all the time, and we don't just want to store them sequentially like a film, but we need to learn something from them and associate? So there are processes there that seem to be doing important things, but we don't know enough about them yet. We don't know exactly why something gets filtered through the hippocampus first and temporarily stored in the medial temporal lobes, and then later on seems to be reactivated multiple times so that it gets stored throughout the neocortex and related with these low-level systems in that way. But it does seem to serve a purpose, and we have some hypotheses, some theories about it, and I think the more that we look at those in detail, you may learn about useful ways of handling the information in the real world, which we are adapted to deal with, that you can then apply, for example, in AGI. We live in the real world. Our brains are evolved to be there. It's not optimized, it's not perfect, there are many things we would wish for it, and an AGI system doesn't need to be like the brain, but I think that we can learn a lot about how we function in this world with this brain as a way to make AGI.
Yeah, we don't have much time, so I'll keep my reply on this very brief. But I think it's a matter of what level of precision you want to look at. Because on the higher level, of course, if you look way back, cybernetics from Norbert Wiener was all about brains and animals and people and machines, and this led to computer science. So the cross-pollination between understanding of brain and understanding of computer intelligence has been there all along, and on a high level is certainly still there. Another analogy I thought of it is walking, actually, which isn't to do with the brain, but to do with people. Because if people didn't walk, we probably never would have thought to make robots walk, because it's a real pain. They would just roll around on wheels. But we observe that people can walk, then we study things about how people walk, and that guides how we make legs and how we make robots walk. And there's certainly, even if you're not trying to make a detailed biological leg, there's a lot that you're picking up from how people walk to make robots walk. And I think a similar thing occurs with thinking. Even those of us who aren't trying to emulate brains in detail, we we're getting general principles from how the brain works as the neuroscientists throw them to us, which guide the systems we build to do similar things, and do things we never would have thought about, looking at digital computers on their own. I
guess there was one last question up there. Then we have
one more question, then we gotta have lunch.
Yeah.
It looks to me that there's a relationship between the biological investigation and the computer AGI programs. There's one program I'm working with where I'm working with concepts and with rules. The concepts represent information from the outside, what the computer gets, and the rules represent what the computer is supposed to do. And I'm now realizing, after your talk, that really the concept can be represented as a neural column or neural field. And the rule, because the concept has an output which would be a binary output which represents the concept. And the rules are also a type of neural field in the sense that there's an input at the rule and the output of the rule also is binary and is an output. So it looks to me that there are many similarities between the human brain and what we are trying to do.
I would agree that there are definitely enough similarities for it to be interesting. Also, what you were just saying reminded me again of a topic that came up yesterday with Richard Sutton, and this was about the off-policy learning. And of course, off-policy learning is something that is perfectly natural for the human brain. And in the context of yesterday's talk, when I was giving a tutorial on how reinforcement learning may be implemented in the prefrontal cortex, well, one of the things I didn't really talk about extensively, but that was present, is that even though my value functions, they're not quite the way that they're expressed in reinforcement learning, but off-policy learning was a natural part of the system, because you have many columns that are representing certain things and that have a way of interacting with one another with sufficient population so that you can create all kinds of different pathways from one column to another, and you can automatically learn these connections, even in a totally different context. If you're handling one task, but you happen to come across interesting associations, these are being picked up, and that's giving you off-policy learning that may fit into a completely different pathway through these mini-columns. So the interesting thing about that part of Richard Sutton's talk about off-policy learning was that off-policy learning wasn't really a problem for the prefrontal cortex model, although trying to express the value of different rewards and how you might, best optimize the action selection and things like that, that's more complicated, or at least we haven't quite figured out everything about it yet. But some things you get as a bonus, you just get naturally if you're exploring the biological system. And that may give you an idea about what kind of an architecture or what kind of representation you can use in AGI. So I think that there are enough things in common, and there are these bits and pieces that you can sometimes get an inspirational idea from. That's why the whole concept of brain-inspired work in AI or in robotics is a hot topic.
Okay, so let's thank the speaker again.
Are you back online?
Architect